Hello and welcome to Streamlining Protocols for Protein Microarrays. My name is Vicki and I will be in the background answering any WebEx technical questions. If you experience technical difficulties at any time during this WebEx event, you may contact WebEx technical support at 1-866-229-3239, or you may submit your technical issue via the Q&A panel and I will attempt to assist you. We do encourage everyone to use the audio broadcasting, but if you'd like to join the teleconference via the phone, you will need to close the audio broadcasting box and request the phone by clicking on the phone icon below the participant list. It will then provide you with the dial-in number, along with the event number and your personal attendee ID number. We will be holding a Q&A session at the conclusion of today's presentation. You may ask an online question at any time throughout the presentation today by simply clicking on the question mark icon located on the floating toolbar in the bottom right side of your screen. Please send questions to all panelists. Today's webinar is being recorded, and a link to the recording will be made available to everyone through their email. With that, we invite you to sit back, relax, and enjoy today's presentation. I would now like to introduce you to your first speaker for today, Sandra Priest, Product Manager for Microarray Systems at Molecular Devices. Sandra, you now have the floor.
Okay, thank you, Vicki. So welcome everyone again, and thanks for joining the webinar, Streamlining Protocols for Protein Microarrays. This is a joint webinar between Grace Biolabs and Molecular Devices to help you optimize and make the most of your microarray workflow. We are very pleased to have you with us today, and I can promise you it will be very exciting and packed with lots of insights and new information for you. Before I let you go off with Michael Schultz, Director of Research at Grace Biolabs, I would like to take a couple of minutes to introduce you to the Dreampeak microarray system and highlight three of the key advantages for your protein microarray research. The Dreampeak microarray system is a fully integrated system consisting of microarray scanner, all-in-one software, the Dreampeak Pro, to control the hardware as well as analyze the images and extract the data for further downstream processing. For the bioinformatics side of things, we have the Acuity software, which excels at analysis across multiple microarrays as well as data warehousing. If you would like to get into more detail about optimizing your software and data handling, stay tuned. We'll shortly offer more webinars on this topic. Let me go to the next slide. We take a little pride in continuously finding the best solution for current and evolving microarray needs to offer a state-of-the-art microarray scanner, as you can see on the slide. For over 13 years now, our customers' loyalty proves that we are doing the right thing. So that means you can't go any wrong with the Dreampeak microarray scanner. We have several thousand scanners in the field. That means in every third lab, microarray lab, we have one. So you might as well have one on your own, or you may know someone who owns one of these little blue powerful machines. On the next couple of slides, I'll demonstrate a few of the key advantages of the optical and mechanical system that will be such a benefit for doing protein microarrays. I'll step into the debate of a confocal versus a non-confocal system, show the effect of how the slide is mounted in the scanner, and give you an idea of the incredible flexibility of the system with user-accessible filters to expand your options. So there's always a debate if a confocal or non-confocal system is better for scanning microarrays. On the left, you can see a confocal system like it is being used in a microscopy and as well as in some microarray scanners. With such an optical system, you reject light that is outside the plane of focus, that is displayed by the slices through the ball on the left. This is useful if you have samples that are three-dimensional in shape, but it is absolutely not necessary for microarrays. It even can turn out to be a huge disadvantage, as you will see in a moment. So the best system for microarray scanning is using a non-confocal optics. As you can see on the right, and you'll know that actually, the microarray spots are in the same focal plane as a background. There is just no way you can optically slice through spot and background. But, and this is a big but, with a non-confocal system, you will collect a thicker slice and collect even more fluorescent light from your sample. So if you want to choose the best system for your protein microarray, you'll want to go with a non-confocal scanner. On the next couple of slides, I'll show you the effect of the slide mount in play with a confocal or non-confocal system. So we are seeing a slide mounted on its back and on its non-printed side in a confocal system. Slides, even though they look extremely flat, if you have them on your table, they're not perfectly flat. They have slight surface unevenness, like warps and tilts and even mountain-like structures. So with a confocal system, you will run in focus and out of focus as you scan across the surface. And you can see this in example if I scan through the slide a couple of times. Here we have the mountain-like structures. It will be in focus and out of focus and in focus and out of focus. So the green, as you can tell, shows the laser beam and the narrow depth of focus. So it's actually not meant to be like this, as the image will, if you scan it, will get brighter and darker as you scan. That is not a problem of your microarray. It's actually a problem inherent to confocal optic systems. There is nothing you can do about it if you have a scanner like this with this kind of setup. Let me go to the next slide. In the Dreampeak system, slides are mounted on the spotted side, and that is on its face, so to speak. This already compensates tilted and warped slides. And then you have a way broader range of depth of field. We have, for example, we have 64 micron as opposed to 1 to 5 micron in other scanners. So you can see this as a thick green illuminated field in the animation. And let me just run through the animation so you can see it is scanning through. And even with the mountain-like structures, you'll always be in focus. So you'll all capture the light that is there. You're collecting light from the steep area, and therefore it will be always in focus. And your signals will be very even. And actually, I assume you'll be interested in the highest data quality possible. So because of that, you'll probably want to go with a non-confocal scanner for your protein arrays. And I know that there are a lot of problems and issues when you first start out dealing with protein microarrays, and you don't want the scanner to be part of this. Lastly, I want to, Victoria, can you advance to the next slide, advantages, full access to optical filters? Lastly, I want to highlight another key feature of the Dreampeak system, that is the user-accessible filter wheel with up to 16 filters. For protein arrays, when playing with different dyes, you'll want to select the optimal filters to gain as high and clean fluorescent intensity as possible. Exchanging filters is easy as 1, 2, 3, as you can tell from the picture. And you can buy all kinds of special filters either from us or from third parties like Samrock or Chroma. They have very high quality filters. And in one of our next webinars, we'll address the use of quantum dots, the blue laser and a nice set of filters will really show their importance for that, and certainly expand the capabilities of your multiplex assays. Okay, so I'll now go on and we'll come to the main part of today's webinar about streamlining protocols for protein microarrays. And I'm very pleased to hand over the presentation to Michael Schultz, Director of Research at Grace Biolabs. And again, we'll have some time at the end for questions and answers. So, Victoria, can you pass over to Michael, please? I think I lost the connection.
Yep.
Thank you.
Thank you, Sandra. I was just advised by our personnel here that as you ask questions, go ahead and ask them throughout the talk, and then we'll answer them at the very end. So welcome, everyone, and thank you for joining us today for the first of these 3 webinars geared towards obtaining optimal data with protein microarrays. So today we will cover some of the aspects of performing protein microarray assays, from choosing the appropriate binding substrates to choosing detection methods, as well as other considerations which can impact the data generated with protein microarrays. We will be addressing these topics today in the context of the microarray workflow and paying special attention to various parameters throughout this workflow, which can really affect your microarray results if not properly optimized. I'll provide examples from data that we've generated through our efforts here at Grace Biolabs aimed at minimizing array data variation and improving signal-to-noise and assay dynamic range. There are likely many of you joining us today who have not had any significant exposure to protein arrays, and hopefully this will serve as a building block towards your successful implementation of this technology. And for those of you with prior microarray experience, I'll provide examples throughout this talk which may address some issues you are currently experiencing and hopefully provide some insight. Oh, the slides aren't progressing.
Go ahead and click on the slide deck and then use the arrow buttons on your keyboard.
Can you click on this? Just click on there, on the slide deck. There we go. So I'd like to first tell you a little bit about myself and my background. I've worked for Grace Biolabs now for 2 years, where I am currently the Director of Research. I obtained my doctorate in pharmacology and toxicology from UC Davis and have held various positions using both DNA and protein microarrays in the last 12 years. I've worked with the arrays in the capacity of an end user as well as a provider of this technology in both academia with my work, postdoc work, and array core microarray facility work, and in industry as well with various biotech companies. So I attended the International Human Proteome Organization meeting in Boston last week, and the data presented by a variety of researchers in systems biology was really impressive, and the power of many high-throughput proteomics techniques was apparent. But it was clear from Rudy Ebersold's talk on the HumanProteome Project, these techniques have yet to be fully adopted into the research mainstream. The reality is that there are barriers to implementing high-throughput proteomics methods like mass spec and microarrays, and it's reflected by the fact that many researchers still rely heavily on the Western blot and the ELISA for protein-related research. But protein microarray technology is becoming more accessible, and researchers are beginning to benefit from the multiplex capabilities of this technology. With relatively few modifications to current ELISA and Western blot methods, it comes the ability to assess more than 1 or 2 proteins at a time, and allowing for a whole or focused proteome analysis in single experiments. With this also comes the advantages of small sample requirements and reagent usage, which should translate to lower costs of your assays. Now, there are various configurations for protein microarrays, and these can be categorized into two basic types, forward phase or reverse phase, with forward phase being the most common. Forward phase arrays typically are assays where purified molecules are deposited on the array and they're probed with a complex sample. The purified molecules can be antibodies, as depicted here, purified proteins, peptides, or DNA, or any other small molecule. An example when using antibodies would be for a capture assay. The captured antigen can be detected in various ways, such as after a sandwich assay, much like for an ELISA. Peptide or purified protein arrays vary, but example applications include the detection of autoimmune antibodies in patient sera, or for high-throughput screening of protein binding motifs to assess protein-protein interactions. Another configuration is termed nucleic acid programmable protein arrays, or NAPA, where DNA expression vectors are spotted and proteins are expressed directly on the array spots. And possible applications for this configuration vary, but they have been used to study protein-protein interactions and protein small molecule interactions. Another type of configuration, which is increasing in use, are termed reverse phase protein arrays. In this configuration, cell or tissue lysates are directly spotted on microarrays, and various samples can be probed for individual biomarkers. This technique was developed for the identification and the study of prognostic biomarkers for cancer, where samples such as tumor biopsy lysates from many patients can be screened, spotted at once on one array, and probed with individual antibody biomarkers. So it's clear that there are many formats for using protein microarrays. So when starting out, it's very important to first define how to configure a microarray for your specific application. And considerations include what your sample is, whether they're purified molecules or complex mixtures, what your spotting material will be, what your assay requirements are, and how you will detect your signal. So how you detect your signal is an important parameter to consider early on in the process, and there are various compatible with microarray applications. The two most common platforms for protein arrays include fluorescence and colorimetric detection. On the left are typical image data collected by fluorescence detection using visible or near-infrared dyes. These images were obtained with fluorescently labeled secondary antibody, much like for a fluorescent ELISA assay. These would be detected with a fluorescent microarray scanner, such as the Dreampeak 4400 shown here. On the right is a typical image from data collected by colorimetric detection, as would be used for methods in immunohistochemistry. In this case, the data is from a horseradish peroxidase catalyzed reaction with diaminobenzidine as the precipitate. And these arrays can be imaged by various methods, such as with a standard flatbed scanner. So you'll want to choose a detection method which is appropriate for your application. So for example, here is some data contrasting the signal dynamic ranges for fluorescence and colorimetric detection. Shown are serial dilutions of IgG, which were microarrayed versus the relative signal obtained by the two methods. Although colorimetric methods can utilize relatively inexpensive instrumentation, they are very limited in their dynamic ranges. So this limitation can be overcome with optimized exposure times and assay conditions. But this process can be lengthy, but once optimized, can yield pretty robust data. On the other hand, fluorescence detection can have higher upfront costs for the scanner instrumentation, but has significant advantages in signal dynamic range and sensitivity compared to colorimetric. Once you've defined the configuration of the microarray format and the detection method which works best for your application, it's important to consider various aspects within the microarray workflow to obtain the best data possible. At this point, I would like to address some considerations which can impact overall data integrity from protein microarrays. I want to discuss these in the context of the microarray workflow, which I've organized into three general categories: the preparation of the array, performing the assay, and data analysis. Each of these categories contains multiple steps which will require thorough optimization, without which significant variability can be introduced into your experimental data. An important parameter is to consider the binding surface for your microarray assays. And there are various substrates available which are widely used for protein microarrays, but it's important to consider which surface is most effective for your application and your detection method. Common surfaces include functionalized planar glass. For example, these are epoxy or aldehyde silane chemistries on regular glass microscope slides. Other coatings include thin films, such as non-porous nitrocellulose. In addition to these two-dimensional surfaces, there are various three-dimensional surfaces for proteins, and these include various hydrogels and porous nitrocellulose film slides. Each surface has strengths and weaknesses, and it's important to consider what's important for your application. Again, high binding capacity is desirable for many applications, as you want to maximize the possible binding sites for your antigen of interest. For example, reverse phase microarrays require the highest binding capacity possible for detection of very rare biomarkers. It's important to assess how you want to attach your proteins to the surface, whether it be covalently or non-covalently. And for example, covalent attachment is commonly used for peptide microarrays to orient the peptides in the proper configuration. Your detection methods should be considered when choosing substrate materials as well. For example, fluorescence detection will require minimal background fluorescence, and a white background, such as from porous nitrocellulose, is highly advantageous for colorimetric detection. So there's some parameters you want to consider there for your detection method. Additionally, if specific spotting or binding buffers are required for your application, which is the case for some users, it's important to consider the spot size and the morphology of the buffer surface combination. So for example, some high detergent containing buffers, such as from lysis buffers in RPPAs, form pretty large spots on most functionalized glass surfaces, which will impact spotting density as well as dilute your signal from those spots. There we go. So I want to take this discussion of binding surfaces further with some examples of how these parameters can affect your downstream data. So I will use porous nitrocellulose as an example because its high protein binding capacity makes it ideal for many protein microarray applications. So these data compare protein binding capacity for porous nitrocellulose film slides and other functionalized glass slides. Serial dilutions of IgG were spotted on each surface, as shown on the x-axis, and they were detected by fluorescence, and shown here as relative fluorescence units on the y-axis. Functionalized glass slides include silane chemistries and hydrogels. Performed similarly to each other. Their protein binding capacities were approximately 1/10 of a microgram per square centimeter, and one can see loss of linearity at around 100 picograms of IgG. Protein binding for porous nitrocellulose film remains linear in excess of 2 orders of magnitude more than the functionalized glass slides. Its binding capacity is roughly 400 times higher at around 40 micrograms per square centimeter. So in a few slides, I'll show how high binding capacity in combination with other surface properties can yield optimal results with protein arrays. So what gives porous nitrocellulose this high binding capacity is its extremely high surface area because of its porous structure. The seen here is an electron micrograph of a film slide cross-section. This thick, sponge-like surface of nitrocellulose films can allow for protein binding throughout the entire film depth, compared to a planar glass surface on the right. In addition, the surface is highly irregular by nature, allowing uniquely folded proteins to find optimal binding sites during spotting. Binding occurs through numerous weak interactions at various points of contact with the folded protein. In practice, this provides very effective binding and promotes an environment for optimal protein folding. So in addition to high protein binding, for fluorescent endpoints, it's important to consider the background fluorescence of the binding substrate. So here's an example of showing how porous nitrocellulose film slides are not all equivalent and how high background fluorescence can greatly hinder the ability to detect low abundance signal. A dilution series of total serum on the x-axis was spotted on supernova porous film slides and 3 other porous nitrocellulose film slides. These arrays were assayed with anti-IgG antibodies and signal detected by fluorescence, as shown as relative fluorescence units on the y-axis. On the right are corresponding fluorescent images for the different slide types, showing the variability in background between slides and their corresponding effects on signal to noise. So you can see with the supernovas, we're looking at about 25 times higher signal to noise than with the other slides, just based on the background alone. The high signal to noise allows for a broader dynamic range of signal, as you can see on the lower end. This allows for the detection of lower abundance proteins compared to the rest. So why are these parameters important? Ultimately, a microarray assay will benefit most from the highest signal-to-noise possible. So the combination of a high protein-binding surface with low autofluorescence allows for the most robust signal-to-noise in your functional assay downstream. So shown are signal-to-noise data obtained with supernova porous nitrocellulose film slides compared to silane chemistries and 3 other 3D hydrogel surfaces. Data are shown as the average signal-to-noise for 6 different cytokines detected with specific anti-antigen antibodies on the different surfaces. Because of the surface properties of the supernova porous film slide, it provides 3 to 10 times higher signal-to-noise compared to the other binding surfaces. So once an appropriate substrate has been chosen, it's also important to consider how the protein content is printed. It is important to consider a spotting buffer, as this can directly impact binding and spot morphology. Spotting humidity can also affect spot size, as well as source plate evaporation. Source plate temperature should be kept as low as possible to minimize protein degradation during long spotting runs. Post-spotting incubation should be assessed for each application and may be necessary for optimal binding to some surfaces. For example, NHS ester chemistries typically require an overnight incubation at high humidity to complete the binding reaction. In addition, storage post-arraying should also be assessed, as labile proteins may lose immunoreactivity during storage. I won't discuss this today, but I will explore this topic in more detail in our 3rd webinar of this series. So here are some examples of how spotting buffer components can impact protein microarray results. On the left are images of spotted proteins in various spotting buffers. 1xPBS is a common spotting buffer used in many protein arrays, but when supplemented with different salts and detergents, one can modulate the degree of protein binding as well as spot size. One can clearly see the effects on spot size as well as signal intensity based on the heat map down below. On the right is an example where an in vitro translation buffer is used as a spotting buffer. During long spotting runs, evaporation of source plate solution can be a problem, like I mentioned in the previous slide. In this case, it leads to an inadvertent concentration of critical buffer components, and it results in inconsistent spot morphologies and signal. Ultimately, the result is unwanted variability in your data. So moving along our microarray workflow, much variability can be introduced by assay-dependent parameters as well. Many of these are assay-specific, depending on how your assay is performed, and will require optimization. These steps range from how samples are collected to the choice of reagents used during the assay. Sample collection in most cases requires control of temperature and inclusion of reagents to prevent protein degradation. For example, it is recommended to include protease inhibitors to sample lysis buffers. In cases when assessing phosphoproteins, phosphatase inhibitors should be included as well. Reagent choices can vary widely with application, but one substrate-specific reagent which we can discuss today is the choice of blocking reagent. Again, I'll choose porous nitrocellulose film slides as the example, but this is important for choosing the right buffer for all binding substrates. In the case of nitrocellulose, many protocols call for Western blot blocking conditions and reagents to block the nitrocellulose film microarrays. And for many applications, these reagents are just suboptimal and can lead to low signal-to-noise of the arrays. So due to its high binding capacity, porous nitrocellulose film slides require thorough blocking, especially for fluorescence applications. Use of an optimized non-protein blocker optimized for the substrate can greatly improve the data produced. On the left are typical images obtained from two-color fluorescence assays blocked with Super G blocking buffer, which is a non-protein blocker, versus standard PBS with tween blocking. And note the extremely high background for the green channel with PBST. Data on the right are shown for two excitation wavelengths, on the x-axis, the 532 nanometer and 635 nanometer wavelengths, and are expressed on the y-axis as background relative fluorescence units as a percentage of PBST blocking, and that's represented at 100% by the red dashed line. At 532 nanometers, backgrounds after Super G blocking are 4 to 10 times lower than those obtained with other blockers. At 635 nanometers, backgrounds are up to 8 times lower. These lower backgrounds yield significantly better signal-to-noise. In addition to lower backgrounds, use of a non-protein-based blocker is also beneficial when considering potential cross-reactivity of the detection antibodies with the blocker. So shown here are data from various reverse-phase microarray assays, probing for two prognostic cancer indicators, phospho-mTOR and phospho-P38 MAP kinase. These arrays were blocked with a common casein blocker or with Super G. In the case of mTOR, the data are completely obscured when casein is used as the blocker due to the very high background. The data is similar for the P38 MAP kinase with signal-to-noise, where it's significantly diminished with the casein blocker. In both cases, the use of the appropriate blocker for this surface improved the data considerably. Of course, many other assay-specific conditions need to be considered for optimal results. And these include such things as proper choice of antibodies with the necessary specificities. Additionally, choice of incubation buffer and incubation times should be assessed for optimal results. Another general but often overlooked parameter is the choice of incubation chamber to use during your assay. There are many available chamber options for users to perform their protein array incubations. You'll probably all recognize the variety of options shown on the slide. They range in volume requirements, cost, and effectiveness. Each has strengths and weaknesses. For example, incubations under a cover slip require very little sample, but are limited to simple diffusion during the incubation, and this can really hinder your results. Staining jars allow for ample mixing and are excellent for slide washing, but are not ideal for incubations due to the excessive sample dilution. Automated systems, such as the one shown here, allow for excellent control of incubations and washing, but at a premium cost. Alternatively, there are various adhesive-based seals and clip-on chambers, which also provide active mixing and allow for small sample requirements to be met at a low cost. So in the next slide, I'll show the importance of active mixing during incubations and its impact on data variability. So identical microarrays were processed under a cover slip or in a ProPlate clip-on chamber, which allows for active mixing when used with an orbital shaker. Replicate spots were distributed across the slide surface to assess signal uniformity with and without the sample mixing. On the left are three-dimensional plots of spots obtained after active mixing and one obtained with no sample mixing under a cover slip. There are clear morphology differences between these spots, which are reflected in the data on the right. The coefficient of variations are plotted here and are compared for pixel data within spots, across spots, and on the same array. On the same array and across spots on multiple arrays. Variation was significantly lower for samples obtained with active mixing, giving more uniform spots within and across different arrays. So at this point, I'll return to our microarray workflow. Data analysis will be covered in detail by Sean Carriedo of Molecular Devices in our next webinar. But briefly, I wanted to touch on this aspect of protein microarrays, which really should be considered while you're printing your microarrays. Thought should be given to what microarray controls you'll want to be using, and they should be implemented prior to beginning experimentation. For proper interpretation of results, it's highly recommended to include controls at various phases of the microarray assay process. Positive and negative spotting controls are very valuable and should be chosen to represent proteins normally and not normally present in your sample types. In addition, I find it useful to deposit the positive controls as spatial controls as well, as seen on this image on the right. This can be really helpful to help orient your spot finding algorithms during your analysis of your array data. Spike controls should be considered for use. They are really effective for monitoring sample prep methods as well as assay conditions. These can be introduced prior to sample prep as well as just prior to starting your assay. All of these controls can be very useful for assessing sample and assay quality and for troubleshooting irregular results. I'm sure Sean will cover these in greater detail in the next webinar. So at this point, I would like to summarize the topics we have covered today and how they can impact protein microarray variability. Proper attention to these parameters we have reviewed can greatly increase signal-to-noise and consistency of your assays. Choice of printing substrate has a direct impact on protein binding capacity and of signal obtained from your arrays. Additionally, substrate properties such as background fluorescence can have significant effects on dynamic range of signal obtained with your experiments due to diminished signal-to-noise. Ultimately, it is a combination of optimal binding and background levels which allow for maximal signal-to-noise. Reagents should be chosen with your substrate in mind, as well as your sample type. Optimizing the spotting buffer can have significant effects on spot size, morphology, and signal. Additionally, proper choice of blocking reagent has a direct impact on signal to noise and can mean the difference between usable results and a completely ineffective assay. Lastly, active mixing during your assay incubations enables consistent microarray results within and across slide replicates and warrants careful attention. So this concludes today's webinar. I would like to thank all of you for your attention and hope you have found this information useful. If you would like more detailed information, we welcome you to visit us at gracebiolabs.com. There you can access our on-site guide to microarrays, which covers many of the topics that we reviewed today. We also have a variety of resources available for choosing substrates and chambers that are appropriate for various protein assays. In addition, research results pertaining to various aspects of protein microarrays are regularly updated on our website. Thank you all again, and at this point I would be happy to answer questions that you may have. So here are some questions that came in while I was giving the seminar. Are there companies using confocal scanners for gene and protein arrays, and what is the advantage, disadvantage of confocal? I believe there are some companies using confocal scanners, at least for protein arrays at this point. I believe the TCAN system is confocal, and we know some customers that are using that. Really, in my opinion, there are disadvantages to using confocal from the data that I've generated, comparing the imaging system of the Dreampeak versus the confocal system. Sandra pointed out earlier on in the first few minutes that non-confocal plane or depth of focus is really important for porous nitrocellulose films. We have a film that is 10 to 12 micrometers thick, and so that depth of focus allows you to obtain the data, the fluorescence data throughout that spotting surface. So you really maximize the amount of signal when using a non-confocal system like the Dreampeak. Sandra, do you have anything to add to that?
Well, it's a great answer, and it's also a very good concern of the attendee that asked about the confocal system. It is really very important to have the right choice of scanner or the right optical system to read out the signals, so that you're not running into the problem with uneven data or intensity that drops across the slide. And I think there are mechanisms to compensate for that, but it is for being in the same plane of focus, not being able to dissect the signal from the background on a microarray slide. It is very important to have a depth of field as you have, or as we have in a non-confocal system. So I think that should be answer enough. And you have the attendee has our email addresses. So if you want to follow up with it, or if you have more questions, then just don't hesitate to contact us, and we can give you more information on what to watch out for. And if you'd like to have or to test drive a Dreampeak system in your lab, then just contact us or contact me, and we'll arrange that so that you can run a test with your microarrays on site in your lab and give it a really rough test, so that you're sure that you have the very best system for your protein microarrays. Just let us know, and we'll just come to you and bring a system with us, bring a scanner with us to your lab, and you test it.
Let's see. Next question that was asked was, what were the chemistries compared to porous nitrocellulose for the dynamic range experiment shown? There are a variety of silane chemistries. There are aminosilonized, epoxysilonized, and aldehyde silanes, as well as 2 different hydrogels, both using NHS ester chemistries for attachment. And this kind of addresses the next question, and comment and question, where aldehyde and epoxy chemistries are not the best chemistries for immobilized proteins. Did you compare NHS or DDS chemistries? So, yes, we compared 2 NHS ester chemistries in that comparison. Let's see, looking back on some other questions. Did you compare cross-reactivity of porous and glass substrates? No, not really. Cross-reactivity to the blockers, I presume, is the question. In that sense, no glass was performed, just primarily because Super G is not a good blocker, as far as we know yet, for glass, planar glass surfaces, especially functionalized surfaces that are going to require specialized blocking reagents to inactivate those active sites. Another question was, what are the disadvantages to using split pins? Other than potentially drying effects during long spotting runs, that could be an advantage or a disadvantage of using split pins. In terms of spotting on nitrocellulose, there can be some disadvantages if the spotting system is not finely tuned and the pins are, in essence, being jammed down into the surface during spotting. That will leave little divots in the nitrocellulose. So it does require some optimization of your Z-axis component for the spotter. Typically, that's not an issue for a lot of spotters that use like the stealth technology that, in essence, are not really spring-loaded, but the pins bounce up and down during the spotting, so it really minimizes that effect on the nitrocellulose. Another question: do you suggest us to block the slides for Cypher Ruby staining, which Grace Biolab slides use porous nitrocellulose? So, hopefully you heard that. I was kind of leaning away from the microphone. I'll read that question again. Do you suggest us to block slides for Cypher Ruby staining, and which Grace Bio slides use porous nitrocellulose? 3 of our slide types, the Supernova, the Nova, and the Avid, are porous nitrocellulose film slides, and the Path slide is nitrocellulose, but is non-porous, so they will appear clear and reflective. It's a 2D surface. And do you suggest using the blocking for Cypher Ruby? Cypher Ruby can be performed with and without blocking. I know many users who do not block before doing Cypher Ruby staining and obtain results that are to their satisfaction. Do you know of any low evaporation array buffers for proteins? Typically, a lot of people will put glycerol in their spotting buffers to minimize evaporation during spotting. It really does depend on the substrate as well as the sample type. Some people use DMSO, betaine for controlling evaporation, but it really depends on the compatibility with your substrate as well as your sample type. So, for example, DMSO is not compatible with porous nitrocellulose films, and you wouldn't want to include that in your spotting buffer, but it is very commonly used for glass or planar glass substrates. So the answer will vary, and it really does depend on compatibility. Do you recommend active mixing during blocking stage for protein microarrays? Is there a preference for, it moved on me, for drying a blocked microarray prior to assay or immediately assay after blocking? For porous nitrocellulose, I do not mix during blocking, primarily because as you start agitating the microarray and the unbound portion of the slide is not coated or blocked, you will start seeing artifacts of protein that had not bound to the surface and starts drifting away from the spot. So I typically block under static conditions to prevent any artifacts, and then I start agitating after blocking. And I do recommend that you do not dry slides between blocking and assay. You want to keep your slides wet during the whole process, unless you've added a preservative blocker combination and you plan on storing your slides after blocking. We observe different signal-to-noise ratios in different blocks of slides. What would cause this? Any way to make it more uniform? That, to me, typically relates to active mixing during your assay. Without more detail and seeing images, it would be tough to guess as to what's going on there. But typically, active mixing remedies those types of problems. And for the previous question, the signal is pretty uniform. Okay. That changes things. That could be related to blocking. If signal is uniform, but the background is inconsistent, that would definitely affect your signal to noise. So it could be inconsistent blocking of the surface. Again, there's so many individual variations and experiments. I'd have to look at images. So you're welcome to send sample images to me directly, and I could actually discuss this with you offline. And my email, I don't know if it's there it is on the page, mschultz@gracebio.com. Feel free to contact me with specific questions. So I think that pretty much covers all the questions that are on the board. And again, I'd like to thank everybody for joining us today. It's been a pleasure being here, and we look forward to seeing you again on the next webinar with Sean Carriedo, discussing data analysis, and then I'll revisit again in the third webinar to discuss additional topics concerning protein microarrays. So thank you, everybody.
Ladies and gentlemen, thank you for attending today's webinar. You may now disconnect your line. Thank you, and have a nice day.